Today we're going to discuss the cytoplasm of the bacteria. So to just get into more details, so as a definition, the cytoplasm is a viscous watery solution or soft gel that contains the variety of organic and inorganic solute. It's neutral, which means that it's pH is 7. It is rich in enzymes important for metabolism, as well as we have the um, as an example of the enzymes, we have the DNA polymerases, RNA polymerases, and others. And of course, we have the water, which is the matrix where the components interact with each other. We have the mRNA and the tRNA, etc. There are no organelles in the prokaryotes, as we just said, and as we said in the previous videos. And of course, we have an exception that we have ribosomes. These ribosomes, they can either be free alone or they can be clustered in what is called polysomes. We also have in the cytoplasm an irregularly shaped region called the nucleoid that contains all or most of the genetic material. This genetic material is double-stranded DNA molecule, circular and supercoiled chromosome. It is condensed by specific proteins. They are not called in this case histones, and as a result of this, because there is no nucleus, so and no organelles except for ribosomes, so the transcription process, the translation process, and the replication process, they all take place simultaneously in the cytoplasm. Just to note that we have in prokaryotic cytoplasm may contain also an extra chromosomal genetic material called plasmids. This plasmid is physically separated from that of the chromosomal DNA and replicate independently, and it is double-stranded circular DNA molecule carry few genes that can confer advantages to the host cell or advantages to the bacteria by itself because it's a host sometimes. Let's say, for example, if somebody takes an antibiotic, this bacteria can have few genes, such as the antibiotic resistance genes, that can lead to the formation of certain enzyme that will degrade the specific antibiotic. So, in this case, the bacteria may hold resistance against this antibiotic, which is advantageous for the bacteria. And we just know that this resistance can be transmitted from a bacteria to another. And this, how can this happen? So we have a process called conjugation that can happen between two bacterial cells through the sex pili that can transmit this plasmid found in the first bacteria to another one. And then this bacteria will have the gene of interest that will start to synthesize enzyme that will give resistance against the certain specific antibiotic. So this is how the resistance will actually be transmitted from one bacteria to another.